Hi, Michelle Glass here. Our job for today is to talk through, let me see, Lab 11, and I want to go through Activity 1 with you. Now, if you need a little bit of extra background information on Mendelian genetics, dominant and recessive alleles, I've linked some other videos for you on your D2L page, so make sure you're looking at that background information and reading through the background information in your lab. This video is going to talk through how to tackle Activity 1, monohybrid cross, and the chi-square analysis. So make sure you have your lab with you so that you can work through those pages. And then you're going to be working through Activity 2 with just a little bit of help from me on video. So let's get started. Okay, so to begin, we're going to be looking at a heterozygous parent. So let's double check what we mean by heterozygous. Heterozygous, that prefix hetero means different. So we're looking here at, in this case, corn. We're looking at kernel color, and we're going to say that these parents are both heterozygous for corn color. And so that means we're going to give them both a dominant and a recessive allele. And our lab manual just has us assign the letter A for the gene for corn color. So the capital A is the dominant allele, which in this case is purple color, and the lowercase a is the recessive allele, which in this case is yellow kernel color. Now both of our parents are going to be heterozygous. So what we're going to do next is trace through meiosis to figure out what type of gametes each parent can produce. Okay, so remember, if the parent starts out as diploid, meaning they have a complete set of DNA, before the process of meiosis even occurs, the cell is going to duplicate or replicate that DNA. So here you can see I've represented that. I now have 2 copies of my chromosome with my capital A and 2 copies of my chromosome with my lowercase a. So I'm representing here my sister chromatids. I'm just really simplifying the image. And then we're going to represent meiosis 1 occurring. So meiosis 1 is we're going to see the separation of homologous chromosomes. So we're going to get 2 cells at the end of this. One cell is going to have the capital A set of chromatids, and the other cell will have the lowercase a set of chromatids. And then let's go ahead and represent through meiosis 2. And when we do that, we see our possible gametes here. Even though we're talking corn, we're still going to call these either egg or sperm cells. And you can see that we're making 4. We have haploid cells. And because we either have a dominant allele A for purple color or a recessive allele lowercase a for yellow color, then that's really our 2 types of gametes that can be produced. Now remember, both parents are described in our manual as heterozygous. So that means the male can contribute capital A or lowercase a, and the female can also be contributing the same information. Notice too, there's repeats there. So we can just pull out the gamete that has the dominant allele and the gamete that has the recessive allele. Next thing we want to do is practice our Punnett Square. So if you're needing to draw your own, this is your easy way. Do 3 vertical lines and then 3 horizontal lines. And then in this top box here, you don't really put anything. And sometimes students find that confusing. So what I like to do is represent my female symbol and my male symbol. And that just helps to remind me that that helps to remind me that this set of boxes here is going to be representing where my female gametes are, so my possible egg cells. And this over here is representing where my male gametes are, or my possible sperm cells. Now, it doesn't really matter. These could be the eggs, these could be the sperm. That's just totally random. But that's the reason why I do that little symbol there. So how do we know what to stick for those gametes? We look at this picture here. So remember, our female can be producing an egg with a dominant A, capital A, or an egg cell with lowercase a, which is the recessive allele, and the male is doing the same thing. So we can just plug that information into our Punnett square. Those boxes in the middle of the square are representing possible possible zygote combinations. A zygote, by definition, is a fertilized egg. So this is when the sperm fertilizes the egg cell. These are the possible type of offspring that can be produced, okay? So what we're going to be doing next is taking an egg cell and a sperm cell and showing, okay, if this sperm fertilizes this egg, this is the possible zygote, okay? So you can see that I've put that into that first box there. That first possible zygote is capital A, capital A. Now, the only real rule about filling in these boxes is to always write the dominant allele first and the recessive allele second. And that is going to be the common way that you see these completed. That's going to be the easiest way for you to understand your Punnett square. So it's really your only rule. So let's pause the video and see if you can complete it on your own. So go ahead and pause. All right, so check your Punnett square with mine. See how you did. Good job. Okay, next what we need to do is figure out what is described as our genotypic ratio. So the word genotype is talking about what alleles, what genetic information was actually inherited. So the question here is, are you big A, little a, or little a, little a? When we do big A, we can call that homozygous dominant. Big A, remember, is heterozygous. And little a is homozygous recessive. Now we always write it in this particular order. So anytime you're reporting a ratio or probability, you always want to make sure that you're putting it homozygous dominant, heterozygous, homozygous recessive in that order. So all we have to do is use our Punnett square and count. So we look at our Punnett square, how many are big A, and write that number down. Excellent, so that was easy counting, right? There was just one. Now do the same thing for your heterozygous, which is big A, little a. Good job, two. And then finish it up with your homozygous recessive, which is little a, little a. And that was easy peasy, one. So this is what we're gonna call a ratio, which we can say 1 to 2 to 1, and it's always gonna be, remember, in that order. Or we can represent them as fractions, which would be 1 out of 4, 2 out of 4, 1 out of 4, or percentages. So 25%, 50%, 25%. Okay, remember this is just looking at the genetic information or the allele combination. The second thing we want to look at is what's called the phenotypic ratio. And the term phenotype here means the physical appearance or expression. So what color is the corn? Remember, we have two choices. The corn kernels can be purple or they can be yellow, right? If we look in our lab manual on page 104, it tells us that the purple is dominant, so we've been representing that with a capital A, and the yellow color is recessive, so we represent that with lowercase a. If you have reviewed your Mendelian complete dominance rules, you'll remember that as long as you have one dominant allele or capital letter, then you're going to have the dominant form. And the only way then to be yellow in this case is to have two recessive alleles, which are represented little a, little a. Okay, so count it up. And you should get a ratio of 3 to 1, 75% to 25%, or you can call it 3 out of 4, 1 out of 4. And we always write the dominant trait first and then the recessive trait second. Okay? Nice. Okay, so we're going to do a little experiment. So we have this ear of corn, and we see when we look at this that we're seeing both purple and yellow colored corn kernels. And the lab asks us to go ahead and count 100 total kernels and then tally up how many of those 100 are purple and how many of those 100 are yellow. So that's what we're we're going to do next, I've done that for you. So here is our raw data if we had 4 groups like we would have during class. So here we did the count with 4 different ears of corn, and these are our totals. You can see how many purple, how many yellow. We did 4 ears of corn, 100 kernels per ear, so we counted a total of 400 kernels here. So what we want to do next is learn how to do a chi-square statistical test. And a chi-square test allows us to compare our experimental raw data with a hypothetical model. In this case, our hypothetical model is based on Mendelian genetics and our Punnett Square. So we're really asking the question, is corn kernel color, say that 3 times really fast, corn kernel color, corn kernel color, corn kernel color. Okay, if corn kernel color is inherited through Mendelian genetics, meaning complete dominance, okay? And we're going to be able to compare the data that we just looked at with what we expect based on our Punnett Square. So the next thing we need to do is to think about, okay, well, what do we really expect? What does that mean? So think about what we did with our Punnett Square. What percentage of your corn kernels should be purple if inheritance follows the Mendelian genetic rules? Can you come up with that answer? Look back at your Punnett Square for help. We're looking at purple. Do you see it? Right on, it's gonna be 75%. So now think about that for yellow. What do you think about yellow? Again, check out your Punnett Square. Okay, fantastic, you're with me, 25%. Okay, so we're going to be using these percentages to figure out what we're going to call our expected value. So these are our probabilities, these are our expected frequencies when it comes to our raw data, okay? So your chi-square analysis is a way for you to see if your raw data matches up with your expected model, okay? So let's go ahead and work through the chi-square together. You're going to find this in your lab manual. Let me look up the page. Excuse me. Okay, we're on page 107. All right, so to start your table, you're just filling in the table that's already there for you. We need to put in our observed data. So total, we counted up 302 purple kernels and 98 yellow kernels. And just at the bottom of that column, go ahead and add together, make sure it equals 400. That's always a good check to make sure you're doing your numbers right. Okay, then we need to put in our expected values. Remember, expected is based on our Punnett Square, based on our hypothetical model of Mendelian genetics and complete dominance. So what we're going to do here, we've observed a total of 400, so we expect 75% of those to be purple. So we take 400 times 0.75 and we get 300. And then we expect the remaining 25% to be yellow. So we take 400 and we multiply that times 0.25 and we get 100. So that's our expected value. Okay, now if you're having any questions about how we got those, make sure you send me an email and get some extra help here. Okay, then we're filling in the next column. This is just some easy subtraction. So you take your observed and you subtract your expected. And typically you get some negative numbers in here. So then the next column, you square it, which just gets rid of the negative. Negative times the negative is positive. And then in our final column, we do a division. So we're going to take 4 divided by 300 to get 0.013, and then 4 divided by 100 to get 0.04. You see how we did that? Yay! Okay, and then you add. And so 0.013 plus 0.04 gives us 0.053. Now go ahead and check everything with the calculator, especially if you're a little bit unsure. Go ahead and pause and check your numbers. Okay, this number right here, 0.053, is our calculated chi-square. And it is the whole reason for that table. It's going to help us get the information we need for the next step, okay? So the next thing we're going to do is find what's called a critical value. A critical value is given in a table, also on page 107. In order to use the table, we have to first calculate what's called degrees of freedom. Now we calculate degrees of freedom by taking your total number of categories and subtract 1. So our corn can either be purple or it can be yellow. So it's 2 categories, and we subtract 1. So our degrees of freedom is 1. It's that easy. Okay, then we're going to use our p-value of 0.05. And this is telling us how confident we are in our results. So this is telling us that there's a 5% chance that we've made a mistake, that even though things mathematically look right, they're not really right. But there's a 95% chance that we're accurate. So we can have a lot of confidence in our results. If we did a p-value of 0.01, that means a 1% chance of making a mistake and a 99% chance of being correct. So that's going to give you even more confidence. Okay, so you're looking at your table on page 107. You want to find your degrees of freedom and your p of 0.05. And that's where you get your critical value. So for us, our critical value is 3.841. Okay, so double check that you can find that. So pause if you need to. Okay, this is what we're doing. We're going to compare our critical value with our calculated chi-square. So that green number at the end of our chart is our chi-square value, 0.053. And we need to compare that to the critical value of 3.841 that we got out of our chart on page 107. Now this is how you do the comparison. Okay. In this scenario, our null hypothesis says there's no difference, meaning there's no difference between our raw data and our expected data. That what we thought would happen is what is happening. That our experimental data matches with our hypothetical model. So in this case, it would be saying that the corn color is inherited through Mendelian genetics. In order to accept that null hypothesis, our chi-square value, that calculated green number, has to be less than or equal to the critical value that we got out of our chart. So our chi-square has to be less than or equal to the critical value in order to accept the null. If that chi-square value is too big, if it's larger than the critical value, you reject the null. So that means corn color is derived from some other way. It's not inherited through Mendelian genetics, okay? So look back at your numbers, and the question that you need to come up with now is, do you accept or reject your null?